We have our next speaker, Toren Finkel from Pittsburgh. As I alluded to, I used to be a mitochondrial person. I guess I'm still maybe a little bit mitochondrial, but Toren is, of course, a star in this field. So, Toren, whenever you...
Thanks, Martin. Thanks to the other organizers for this invitation. It's a real pleasure. I love this microphone. I feel like my Beyonce when I'm up here, which is... I'm not going to sing, though. So my lab is interested in the basic mechanisms of aging, as well as trying to develop therapies for aging. And the older I personally get, the less interested I'm interested in finding new drivers of aging and more interested in finding therapeutics. And so in that vein, I was going to talk a little bit about some ongoing work developing small molecules targeting specific drivers we think of aging. Just some disclosures. And so this is all work I've done in collaboration with my colleagues Bill Chen and Yuan Liu. And I'm going to talk about three unpublished areas that we've been working on. But although they're unpublished, I think they're far enough along to think that there's a path forward in each of them. And the first one is use small molecules to modulate autophagy. The second is small molecules to manipulate NAD levels, which we heard about just by Danica and from others already. And the third is using both genetics and pharmacology to begin to develop a set of targets that I think will serve as a rational basis for senolytic therapies. So again, we've been very interested in autophagy as a driver of aging and age-related diseases. And a lot of our enthusiasm for why we think it's important for aging comes from work of others, particularly people who have worked in model organisms like C. elegans, which have defined a number of pathways and interventions that extend lifespan in worms. And using genetics and epistatic arguments, they've been able to show that in many cases, these very disparate maneuvers that extend lifespan all appear to require or induce a change in autophagy as a downstream effector. And so that suggests that maybe autophagy regulation is going to be critical for aging biology. Although one caveat is that these are all obviously in simple organisms. And the question is, does this extend to more complex organisms? And about 10 years ago, we developed a mouse that allowed us to look visually at autophagy in living tissues. In this case, looking at mitophagy in particular. And what we're able to show is that if you look on the young animals, what one saw, in this case looking in the hippocampus, that there's this intense level of mitophagy going on as evidenced by the sort of orange or red fluorescence. And that same area in an old animal really had a significant deficit or decline in autophagic or mitophagic flux in this case. And so I've always been haunted or intrigued by this picture. And the question is, can you begin to develop therapies that prevent that decline in older animals and actually move the animal on the right to the animal on the left? And so when we began to think about what would be a good target to drug in the autophagy space, I think the candidate that emerged at the top of our list is a transcription factor by the name of TFEB. And this also came up earlier today, but for those who don't know, TFEB is a transcription factor that is sort of the master regulator of sort of all autophagy genes as well as lysosomal genes. And it's involved in inducing an increase in autophagy genes and making new lysosomes in the cells. And it's somewhat unusual as a transcription factor because under normal circumstances, TFEB is restrained in the cytosol, bound to the lysosome itself. And a variety of intracellular stresses, including lysosomal damage, cause a release of lysosomal calcium, which triggers a series of post-translational modifications in the cytosol, which now allows TFEB to move from its cytosolic location into the nucleus, which then induces the expression of 400 or so genes involved in autophagy, as I said, or lysosomal biogenesis. And so while the modifications that happen to TFEB that allow it to go to the nucleus are pretty well described, a very talented postdoc in our group, Travis Lear, has found that once inside the nucleus, TFEB undergoes another series of translational modifications that involve first a phosphorylation by a kinase called PAC2. And that PAC2 phosphorylates TFEB on certain residues that creates what's known as a phosphodegron, which is a signal that that protein should be degraded by an E3 ubiquitin ligase. And we've been able to identify that E3 ubiquitin ligase as a member of the Cullen family of E3s, and particularly that uses this protein DCAF7 as a substrate adapter. And so the end result of that is that TFEB nuclear half-life is actually quite short, on the order of 30 minutes or so, because it is rapidly, once it gets into the nucleus, it is rapidly degraded by this nuclear E3 ubiquitin ligase. And so using that information, my colleagues Bill Chen and Yuan Liu and I have developed a small molecule that interferes with DCAF7's ability to recognize and degrade TFEB. And that compound has gone through a series of iterations, but the most advanced compound we call GT300. And this blocks the nuclear degradation of TFEB and thereby extends its biological half-life and then amplifies its biological activity. And some of that can be seen in the following slides, where we treated cells with increasing concentrations of GT300. And you can see at sort of double-digit nanomolar concentrations, we're able to increase lysosomal number in cells. And similarly, working with my colleagues Emily Roca and Tim Greenemeyer at Pitt, we were able to show that when we dose normal rodents, that we can also see an increase in lysosomal content and lysosomal activity, in this case, in a specific region of the brain that they were interested because of their interest in Parkinson's disease. And so this suggests, and other data suggested, that we had a small molecule now that modulated TFEB activity and could, in a way, use to boost TFEB activities in cells and animals. And so obviously there are a number of diseases in which an increase in autophagic flux might be beneficial. And we've tried a number of these models with interesting results and positive results, but I want to just focus on one, which is this a mouse model of age-related macular degeneration that we've done in collaboration with another colleague, Debashish Sinha. And Debashish has created this model of AMD in the mouse, which is somewhat difficult in the fact that mice even don't have maculas. But with that said, this animal recapitulates many of the pathological features of AMD, including sort of subretinal deposits and a loss of retinal function over a 5 to 7 month period. And so, again, I won't go through all the detail, but what you can see here is that is an ERG, an electroretinogram, which measures sort of photoreceptor and retinal function. And what's shown in I gotta see myself, in black is the normal ERG from the animal. And what's shown in blue is the knockout, the conditional knockout that Debashish has that has this AMD phenotype. And you can see that the conditional knockout fed normal chow has significantly impaired retinal function as measured by this ERG. But again, by giving this TFEB activator, as shown in red, we can basically maintain or restore retinal function presumably by increasing autophagic flux in these animals. And so based on this and other data, this compound has actually been licensed to a company that I'm associated with, and that company is set to undergo clinical trials late in 2024 for this AMD indication. So that's, I think, one example of basically using sort of a known regulator of autophagy and beginning to develop small molecules that may specifically manipulate that pathway. So the second example I'd like to give is the ability to manipulate NAD. We've heard, again, a lot about this as a potential way to potentially reverse or slow a variety of aging phenotypes. And as mentioned, there's a lot of data suggesting that using supplements or NAD precursors, one can replenish NAD. But also, as mentioned, these supplements are not drugs, basically. They don't have drug-like properties. We heard some of them times that they're not what you think they are in terms of purity. They have very short metabolic half-lives, unclear distributions. And so we were interested in trying to use what's known about NAD metabolism to develop real sort of pharmacological agents that manipulated NAD levels. And so, as mentioned earlier today, there are a number of NAD metabolism is very confusing and complex, but we focused on the salvage pathway, which I think many people feel is one of the major drivers of intracellular NAD metabolism. And within the salvage pathway, the enzyme NAMPT is thought to be the rate-limiting step for NAD metabolism. And so what we were interested to do is, could we develop a small molecule that binds to NAMPT and activates it, increases its enzymatic activity. Now, as you know, getting a small molecule to bind an inhibitor enzyme is relatively straightforward, but getting a small molecule that increases activity is much more difficult task. So to accomplish this, what we did is leverage this technology that we've been developing, again, with my colleagues Bill and Ewan over the last 4 or 5 years, which is a screening platform that allows us to identify small molecule interactions with a protein of interest. And this technology is very novel and unique, but I think for the purposes of this talk, the technology that comes closest to it is a technology or a methodology known as CETSA, or cellular thermal shift analysis. And what CETSA does is if you take any protein of interest and you heat it, that protein will denature what's known as its melt curve. So it will go from a native configuration to a denatured configuration. And if you do that same experiment, same manipulation, in the presence of a small molecule that can bind to the protein of interest, then it stabilizes that native configuration. And thereby, when you now heat it, the temperature at which it denatures, the melt curve, shifts to the right. And by measuring that shift and the level of that shift, one can determine the affinity, or can define an affinity of that ligand for that protein of interest. And so we have sort of leveraged that concept and come up with our newer version of this that allows us to do this shift in a sort of high throughput fashion. And so we can screen, and we have screened roughly 30 or so proteins of interest using this approach, and we can screen 20 to 30,000 compounds a day in our sort of fully automated setup. And that has given us hope that we can go after a lot of previously hard to drug or undruggable targets. But in this case, the example I want to show you is using NAMPT as a protein of interest and using this screening platform to basically identify small molecules that bind to NAMPT. And so here's an example where we screened 100,000 compounds of our in-house library, a diversity library that we have in-house. And using this library of 100,000 compounds, we were able to identify roughly 300 or so molecules that appeared to bind to NAMPT with a reasonable high affinity. Now, the majority of these compounds either had no effect on NAMPT activity or acted as inhibitors. But about 5 of these molecules acted as NAMPT activators. And some of the evidence to show that they do so is shown here, where we take 3 of these hit molecules and add increasing amounts of them to a cell and show that, as you see in blue, that the level of NAD rises in these cells. And if we take the same cell line and knock out NAMPT, then these compounds are without effect. So we believe, based on this and other evidence, that these small molecules bind and increase NAMPT activity in cells. Now, this is on tumor cells. What about normal, more physiological cells? And so the same effects we see using these hit compounds on things like iPSC-derived neurons. Now, the effects are not huge. These are hit molecules. We've done a significant amount of chemistry to improve their potency and efficacy. But even at this sort of hit stage, we think that this level of increase of NAD may be therapeutic, because in collaboration with my colleague Stacy Rizzo, we've measured NAD, and as many of you have observed, that the level of decline with age or with age and disease is in the order of around 30 to 50%. And we're able to sort of reverse that with these small molecules. And so we're hopeful, and we obviously have a lot to do, but we're very encouraged by the fact that we can get small molecules that bind to NAMPT, increase NAD, and these have true good ADME sort of properties, very high brain penetration in one case, and so we're in the process of advancing these molecules along. And finally, let me just talk about our approach to senolytics. Again, trying to develop small molecules that act as senolytic therapies. And so, again, there are molecules out there that have efficacy in this space, but again, the majority of these are repurposed agents, often from oncology, and their exact mechanism by which they induce the death of senescent cells is not often that clear. And so we wanted to develop a system to identify targets that would act in a rational basis, would make sense as to derive senolytics against them. And so this is work done by a talented MD PhD student in my lab, Shahini, who's getting married on Sunday. So if I can tell you where she's registered if anybody wants to send a gift. So the system that she used is the cell line that we did this. We wanted to do a genetic CRISPR screen to identify senolytic targets. And so we actually started with a cell line that was very useful in this approach. And that cell line is a mouse embryonic fibroblast cell line that has a floxed allele of TRF2 and an integrated property of a tamoxifen-inducible Cre. Now, TRF2, as you know, is part of the Shelteren complex, and it's necessary to cap the end of telomeres. And so the reason the cell line is very good is because the cells grow like crazy under normal conditions, and then when you add tamoxifen, you delete TRF2 in these cell lines. TRF2 over the course of 1 or 2 you delete the gene over the course of 1 or 2 days, the protein gets deleted, then the telomeres uncap, and the cells undergo sort of uniform senescence. And so that's sort of shown here. Again, the addition of tamoxifen on day 0, it takes a few days, but then the cells stop growing, and eventually 100% of the cells senesce, and there's no escape from that senescence. And so we use this model to do what's in genetics is called a synthetic lethal screen. So the idea was we were interested in finding the set of genes that when deleted in a normal cell had relatively no effect, but when deleted in the context of a senescent cell, caused that senescent cell to undergo cell death. Okay, and so the way we did it is to start with this cell line that I just described, infect that cell line with a whole genome CRISPR library, in this case mouse, because they're mouse cells, and then we select impuromycin. So now we have a lawn of cells in which every cell has one gene knocked out. And then we take that population of cells and we split them. So these cells are isogenic. One we let to continue to grow under just normal conditions. The other ones we add tamoxifen to induce senescence. And then we wait, in this case 12 days, and then we did deep sequencing. Okay? So again, what we're looking for is a gene, which we'll call gene X, that has little or no effect in the control cells, but causes the death of senescent cells. So it's this combination, this synthetic lethality, that it's gene X by itself does nothing in terms of viability, senescence does nothing in terms of viability, but the combination of gene X plus senescence is lethal, synthetically lethal. And on a practical basis, what we were doing is to look for guides that were very abundant in the control population and absent or very low in the senescent population. We got a number of hits of potential targets, and I won't go through all of them, but I'll just go through one to show you why I think this is going to be a useful approach to develop senolytic therapy. So one hit that we got was this gene called PARP16. As often the case when you do a screen, you have no idea what any of these hits are until you start looking at them. But PARP16 is a member of the PARP family of ADP ribosyl transferases. So that means it uses NAD to ADP mono or poly ADP ribosylate its target. There's very little known about it. It's unusual in the setting of PARPs families, which are usually either cytosolic or nuclear. PARP16 is in the ER. And what had been known about it is that it appears to be involved in maintaining proteostasis and inhibiting ER stress. And so the fact that it came out in our screen, and given the fact that senescent cells have a high secretory burden, we thought that the synthetic lethality may come about because of a need for PARP16 in the high secretory state of senescent cells. And so we looked at proteostasis, protein aggregation, in either control cells or cells in which we CRISPR'd out PARP16. And what we saw under normal conditions, there was very few protein aggregates in either the control cells or the PARP16 knockouts. But after inducing senescence, what we saw was that there were some levels of protein aggregation in normal senescent cells, but a tremendous amount in the PARP16 deleted cells. And consistent with that, so again, it's a senescence plus PARP16 deletion that results in proteostatic stress. And also, it's this combination that results in cell death. And so here's again the setting of cell viability in either control cells or two different PARP16 knockouts. So as you can see, there's a loss of viability, again, with this combination of both senescence and PARP16 inhibition. Again, this sort of synthetic lethality concept. Okay, so that screen suggests that if you had a PARP16 inhibitor, you could reproduce these same effects that we're doing genetically. And unfortunately, there were no PARP16 inhibitors. There are lots of PARP inhibitors, which clinically are used for synthetic lethality in breast cancer. But we needed to generate a PARP16 inhibitor. So we did the same thing we did for NAMPT. We screened PARP16 to look for binders of PARP16. Again, using 100,000 compounds, we were able to identify a number of binders to PARP16. We developed a pharmacological assay to take those binders and ask, do any of them act as PARP16 inhibitors? And about 20% of the binders were inhibitors. And then we asked, does that PARP16 inhibitor act as a senolytic? And these are sort of hit compounds, but what you can see in the top panel, the PARP16 inhibitor has no effect on cell growth, but it does, at least weakly, kill senescent cells. And obviously we think that we can improve these molecules in terms of their potency and efficacy. And it also suggests that the other hits that we had from the screen would also be potential senolytics, and that we can build sort of a whole pipeline of senolytics using this CRISPR-based genetics and this pharmacology. Okay, so just to summarize, I've sort of shown three sort of very disparate areas that we're working on, but I think outline sort of a rational approach to developing small molecules for aging, either by modulating autophagy through inhibiting TFEB degradation, increasing NAD through a direct activator of NAMPT, or using, as I said, this synthetic lethal screen to identify targets, and then probing those targets with small molecules. So, and just to summarize my summary, I do firmly believe that aging is druggable, and more importantly, hopefully we'll all live long enough so I can prove it to you. Thank you very much.
I think we have time for one very, very short question. Maybe I can start, or I can ask the question. What happened to the mitochondria?
We're not doing anything yet on mitochondria, so that's next time.
All right. I think we will stop now because the food is already here and it's getting cold. So, but thank you so much, Toren. It was an amazing talk.